Saravana Kumar, Sona Sundaram, and Andreas Huben at IPK. And it should advance. Did it advance? We're stranded. Oh, maybe I push this one. Not advancing. I'm wondering why. Or maybe this. Well, let me just gain some time here by telling that the summary of what I'm going to tell you. I'll give you an introduction on, thank you, on the molecular basis of haploid induction through SNH3. I'll talk about mechanism, and then I'll spend some time on how this technology that was discovered in Arabidopsis is turning out to be possible to transfer, but not easy to transfer to other species. And of course, I'm feeling that I'm preaching to the choir here, but just to remind everybody, haploid induction involves the crossing of a plant such as this one, a heterozygous plant, to a haploid inducer, and then extracting a single genome from that cross, and then that genome can be double, and you obtain a perfect homozygote. You know that a perfect homozygote is a requirement for efficient breeding and commercialization of seeds. So you might sell the seed of an homozygote to the farmer, and the farmer is able to grow an homogeneous crop. If you extract the seed from an heterozygote like this one, well, you get something which the farmer doesn't appreciate. So in breeding, it's short-circuited, it's accelerated by the availability of haploids. And the technology I'm going to tell you this morning about is based on this variant histone, SNH3. Just to remind you, histones are part of the nucleosome, and although there are canonical histones, some of them can be substituted by variant histones such as SNH3. And the importance of the variant SNH3 is that it is the epigenetic signal that determines what position of the chromosome will become the centromere. And the place where SNH3 is present in chromatin is recognized, and a kinetochore is built upon. And of course, the centromere is the handle by which we move chromosomes to one pole at the time of segregation. So SNH3 is embedded into the nucleosome, and it has the typical histone structure. There's a globular subdomain here, which is called the histone fold domain, and is responsible for binding the other histones and binding DNA. And then it has this tail that extends out and does other things. So in the case of SNH3, it is remarkable that it evolves very rapidly. If you look at H3, the canonical histone from a human and a fly and Arabidopsis or maize, they're virtually indistinguishable, maybe one or two or three changes all through the molecule. But in the case of SNH3, the changes accelerate rapidly, and when you compare something like Arabidopsis and maize, there are 38 substitutions only in the histone fold domain, and the end terminus is not recognizable except for the very tip and the very end. So one question which I think is related on how this technology can be translated from Arabidopsis to other crops is why is SNH3 evolving so rapidly? There's one explanation that has been provided by Malik and Henikoff, and that is that SNH3 may engender meiotic drive. And the way it might do so is by the formation of abnormal centromeres in which a repeat present normally has changed this sequence, it expands, and it creates a super centromere. Now, this super centromere may interact better with the spindle, and with SNH3 and the kinetochore and the spindle, therefore, and form an aberrant segregation. This is the megaspore mother cell. It's a polarized structure. One pole will give rise to the egg cell, and the other pole gives rise to dead cell. So if the new centromere is positioned always toward the live cell, then you have something which is very good for the centromere, but is not good for the plant. And so the plant wants to counteract this, and in the hypothesis, it does so by mutating SNH3 and stopping this strong interaction. Is this true or not? We don't know. There is good genetic evidence in mouse and in mimulus, but there's no direct data that says the SNH3 interacts better with a certain type of repeat. So in 2010, Ravi and Chan published a paper in Nature. They were at UC Davis, and this paper described the formation of an haploid inducer when SNH3 was altered. And here is, for example, what a diploid looks on the left side and what haploid looks on the right side. There are 5 chromosomes. It's a little smaller for Arabidopsis, and of course it produces flowers that tend to be sterile unless they magically double their genome. But very convenient technology, high efficiency. So there was a lot of hope that this could be moved rapidly into all kinds of crops. Now, the translation to crops has been uneven. So in Arabidopsis, the haploid inducer described by Simon Chan and Ravi has high efficiency, 40% haploid induction. And actually, this has been translated into wheat and into maize, and I'll tell you a little more later, and where it's a decent haploid inducer. And I consider 5% to be a good minimum threshold that you need to have to make the system really workable. But excellent studies that that were carried out competently and reproduced what had been done in Arabidopsis failed to obtain the same high haploid induction efficiency, and you can see here. So let's then step back a second and say, how does the technology work? Now, before I tell you that, Simon, right after this invention, traveled to Africa. He was excited about translating this technology, and he was hoping that it would help breeding crops in places that where crop improvement would make a great impact. Unfortunately, he contracted an infection and died and passed away. So my lab and Ann-Britt lab, who were collaborating with him, inherited this project. But the way the technology works is this. You first have to knock out the endogenous NH3, and then in the original method, Simon and Ravi made a fusion protein that consisted of the NH3 histone fold domain, then fused a different N-terminal peptide here from a different histone, and then fused the GFP. Now this is also referred to as the tail swap, and it's high efficiency. It also makes the plant somewhat sick. We found that there were other ways of doing it. For example, you could take maize SNH3, complement the Arabidopsis knockout, and that made a high efficiency haploid inducer. Or in the laboratory of Ann-Britt, they found that you could induce amino acid changes and small deletion throughout the length of this protein and get efficient haploid inducers. And finally, it was found, I'll tell you more about it, that you can take an heterozygote of SNH3, and that alone will make an haploid inducer. So how does all this work? Well, first of all, one important thing is that these all vary in efficiency. For example, the tail swap is most efficient when is present in the seed parent, and if you put in the pollen parent, you get a tenfold loss in efficiency. If you just have the exchange tail and not the GFP, you go down in efficiency, and so forth. May's complementation is 20%. Missense are anywhere between 5 and 30% if they work, right? So we spent a considerable amount of time understanding how genome elimination worked. And we knew that in a genome elimination cross, like this, an haploid induction cross, you have an haploid inducer, you cross it to the wild type, and then on the other end of the process, you get haploids, you get aneuploids, you get diploids, and you get a lot of dead seed. So it's a deleterious cross, but you get plenty of haploids. So what happened in the middle, we actually didn't know at the start. We figured that probably the chromosome of the haploid inducers would be missegregated. That was a logical hypothesis, but it took some time to show that that was the case. I want to point out a couple of other peculiarities of the system, which is that the outcross is very dysgenic, very deleterious. It's an incompatible cross. However, if you take two haploid inducers and you cross one to the other, it's a perfect cross. Everything is suppressed. Everything is good. No death, no haploids. Everything is diploid, which create a bit of a conundrum. So the question is, how does SNH3 manipulation cause chromosome elimination? Why is the haploid inducer by haploid inducer compatible? And then how portable is this method? How do you move it into a crop? So I'll tell you right here, because I don't have a lot of time, that the mechanism of chromosome elimination is dependent on the fact that the altered SNH3 is recognized in the mature egg during reproduction and removed, we think, by a quality assurance system. It is just taken away, which leaves the haploid induction centromeres weak. They have hardly any SNH3 left, and these poor centromeres are left to compete with the wild-type centromeres, and that's why they missegregate. Let me show you the key evidence, just a couple of data points. So this is an inflorescence of Arabidopsis. Along the axis, there is from the top to the bottom, there's maturation of the egg cell. So if you take and look at the egg cell, you find that at the before anthesis, the SNH3, in this case, the one with GFP attached, is localized on the 5 chromosome in the egg. Remember, it's an haploid structure. But then immediately as the flower opens, those signals are gone. They are removed. Now, this was discovered by Mohan, and if you look at the wild-type SNH3, you find that it is not removed at all. In fact, you can put the haploid inducer SNH3 and the wild-type SNH3 in the same cell, and the modification is recessive, so at that point it is a functionally wild-type, but the haploid inducer SNH3 is being removed while the wild-type, the unaltered one, stays. So that was a very clear identification of the problem. And if you then send this genome into the zygote, then you can look at what is left. And I'm going to show you here the state of the kinetochore. So I told you that at the level of DNA, there's SNH3. It determines, it's an epigenetic signal. It determines the formation of the kinetochore. Then there are a castle of proteins, but let's just look at two. SMPC is a linker protein that's right next to SNH3 and connects to the rest of the kinetochore. And NAF2 is part of the outer kinetochore. It grabs onto the spindle fibers. So first of all, you can find that the altered SNH3 now in the zygote is present only on 5 chromosomes. You would think, well, you may think these are the 5 chromosomes that came from the haploid inducer. No, they are not. They are the chromosome that came from the wild type. Because remember, the GFP tail swap was stripped off, and both the GFP tail swap and the SNH3 are loaded only on the good wild type centromeres. And that's the same for SMPC. It is the same for NAV2. So the kinetochore only forms on the good wild-type centromere. So the model goes like this. At the time of egg formation, the system recognizes the altered SNH3, strips it off, while on the wild-type side, you leave the SNH3 on the centromere. And in the zygote, there are two centromere types. One is epigenetically poor, no SNH3 signal or very little, and the other one is rich. And then at this point, because you go through a bunch of cell division, you need to load these components, and the components are loaded in a completely biased way. They all go to the good kinetochore, to the good centromere. And eventually these chromosomes are left to missegregate. Surprisingly, they don't missegregate with 100% efficiency. They actually tend to hang in there. That's why you don't have 100% haploids. And if they hang in there for a number of cell division, they actually build slowly back up the good SNH3, and they become normal. So why is that? We actually think we know why this is the case. Oh, and I think a bunch of my animation went away. So here, that's a miracle of PowerPoint. But so what I'm trying to show here on the x-axis is that chromatin has different amount of SNH3. So just imagine that in green, you have a bunch of SNH3 present on the right hand. So it's fully loaded with SNH3. And here there's hardly any SNH3. So this is SNH3 loading on the SNH3 presence. And over here is the velocity of the new SNH3 being loaded. It follows a cooperative response. It loads very slow for regions for centromeres that are poor, and very fast for centromeres that have good SNH3 content already. So the rich gets richer and the poor gets poorer, so to say. And if you think about a compatible cross, both centromeres are here. They both get loaded fast. And if you have an haploid induction cross, one centromere set is subject to high velocity loading, the other one to low velocity. Now here's the important part. If you have two haploid inducer, then both are poor and both get loaded at the same speed, and the problem is solved. That is, in the haploid by haploid, the difference is not there, and so the cell waits until these guys get loaded and then goes on. So that explains why the haploid inducer can self efficiently and why it can cross without any problem with other haploid inducers. So in summary, what I've told you is that haploid induction centromeres are weak, can slowly reload SNH3, and here is another concept I want to introduce. SNH3 necessarily interacts with factors that co-evolve. If SNH3 changes rapidly, then whatever interacts with it has to change rapidly as well. And we know that kinetochore proteins evolve very rapidly. So there are factors that we have discovered that affect this response. And here is a consideration from an ecological point of view. If a plant in the wild makes an altered SNH3 through an amino acid change, that plant becomes, once it makes that mutation homozygous, it becomes less capable of crossing with the wild type. So it's actually it's a speciation. We think it's a speciation gene, and natural variants, in fact, do exist. And this takes too much time, so I won't go through it. But let's get back to the application of SNH3 to crops. And the point that we can make here, 15 years later, is that the efficiency in species 1 is not predictive of the efficiency in species 2. And I'll give you some example. For example, a perfect example is maize. And Mohan, who was responsible for all this biochemical analysis, at one point came up with this interesting insight. He said, well, if you take an heterozygote of SNH3, at the time of meiosis, those two alleles segregate. And for example, a knockout allele would go to half of the progeny cells, and the wild-type allele would go to the other half. And remember that the gametophyte undergoes 3 or 2 mitoses according to sex. So if the gametophyte inherits the wild-type SNH3, then everything is good. However, if the gametophyte inherits the knockout SNH3 in this heterozygote, then each cell division will result in a decrease of the SNH3 present in the centromere. You will just run out of it because you cannot make any more. And so Mohan showed that, in fact, then in the heterozygote of Arabidopsis, half of the pollen, for example, or half of the egg have haploid induction property. And this is, for example, shows that half of the pollen generated embryos that have in a cross that have the wild type centromeres, and the other half has the 5 pattern of the haploid induction cross. In addition, in Arabidopsis, it was an haploid inducer. It was a not so good haploid inducer. And so I even told the people at UC Davis, let's patent this. They said, no, it's a low efficiency haploid inducer, and it's not good. Well, independently, the laboratory of Kelly Doe found that in maize, it actually works pretty well. So they patented it. And they should have, so power to them. Anyway, the point is, difference between Arabidopsis, the same exact technology, 0.5% in Arabidopsis, 5% in maize. So I'm going to then introduce to you another interesting experiment, which shows 2 things. It shows that even in a species as close to Arabidopsis as this one, it's not simple to translate the technology. But second, that we still don't understand the whole thing, because something rather amazing happens when we did this. So let me tell you about the crop that I'm showing here. It's a close relative of Arabidopsis, grown for oil. It's rapidly growing. And so we said, let's make an haploid inducer in this crop. There were several reasons why we were interested. But one reason was that we had a rich set of mutants that were generated by tilling. This is before even the time of CRISPR. And so we knew from the laboratory of Van Britt and Sundaram Kuppu that there were a bunch of mutations that would make an haploid inducer. And here they're shown, plotted, interestingly shows seed death versus efficiency of haploid induction. And you can see that you go along here more and more. You reach a critical level of seed death, and then boom, up you go, and you start making haploids. This is the tail swap up here by comparison. But looking in Camelina, we found the mutation that Kuppu had predicted would work. And let me tell you a little bit about the Camelina genome. It's an allohexaploid, like wheat. It has 3 ancestral parents. Is the last unknown common ancestor, which remarkably has my same name. And then 2 of the genomes come from these species called Neglecta. I'll refer to it as such. And 1 genome comes from Hispida. And this is a little more distant, of course, than these 2 here. So we had a nice tilling set. We isolated, in fact, 198 mutations in the 3 homologs, in the coding region of the 3 homologs, high density tilling. And so we extracted the mutation that were suitable. We needed 2 knockout, right, to knock 2 of these out. And then the third would be a Kuppu mutation. So we constructed them. Here's one with a G83-2E in genome 2, and a knockout in presumed knockout in the other 2. Here's a knockout in the first 2 and G172 to R. And these are predicted to be good haploid inducers in Arabidopsis. Long story short, did not work. We didn't get haploid induction, but we got a decrease in genome size. And so we decided to go and follow it up, see what's happening. And here's the classical analysis. We sequence, flow cytometry only gives you so much information. So we sequence these genomes, and what we can extract then is the dosage of each chromosome purely by the amount of reads that we get on each chromosomal bin. So for example, going along here, we have the 20 chromosomes of Camelina, and the first chromosome here is present in one copy because the dosage of reads is lower, and so forth. So you can see the karyotype nicely here. And you notice that the red genomes, which are the neglected ones, seem to be hit more frequently. So we looked at, in fact, hundreds of these, and the answer is yes. We have one genome that escapes haploid induction, just minimal loss of chromosomes, and the other ones that get eliminated relatively easily. So this is surprising. We had never seen this. And in fact, this is, to my knowledge, the first time that we see a clear difference between the behavior of centromeres with respect to the impairment of SNH3. So we actually got excited about it, and we said there's two possible explanations. One is that we have selection, that is, that the genome of Hispida is expressed better, but all chromosomes are lost equally, but because there is better expression on Hispida, there's stronger negative selection on the aneuploids or that include Hispida, and therefore we don't see them. The other explanation is the one that Malik and Henikoff may be right, and maybe there is something about the centromeres of Hispida that make them special. And maybe they bind SNH3 more avidly and make a better kinetochore. So what's the evidence for this? I don't have a conclusive answer, but we have some interesting observation. First of all, it is true that the Hispida genome is slightly better expressed. So this is in one of the original genome paper published. They take all 3 homeologs for every gene and they rank them, and this shows the ranking. And 38% of the time, in average, the Hispida genome, the Hispida homolog is expressed better than the other 2. But it's not a huge difference. Actually, when we look at our own data, our own expression data in Camelina, or reanalyze other people's expression data with different accessions, we find that the difference is there, but is minimal. So we have a hard time believing that such a difference would create such a bias. But we will test it. So we have a way to test it, and is in progress. Well, the other question is then, can SNH3 binding be specific for a certain type of sequence? And so, I used to be skeptic. I mean, I'm a good friend of Malik and Henikoff. I work with both of them. I think their hypothesis is fantastic, but I like to be skeptic until finally convinced. And so the observation that we have, for example, is that May SNH3 binds the Arabidopsis repeats no problem. So clearly, there cannot be an absolute specificity. And the other one, which is also compelling, is that if you take an allotetraploid and document where the respective genome SNH3 is going, you find that there's no selection. That is, for example, in Arabidopsis, there's an allotetraploid made by an ancestral mating with Arenosa. These two have very different centromeric repeats and different SNH3. So you can make antibodies specific for each and see where they go. They go to the same site. This is a result described recently by Karimi Ashtiani in the Huben lab. We had shown that the Arabidopsis one did the same, a long time ago, but they confirmed it for both SNH3. So no specificity there, right? So we decided, well, let's look at Camelina. Let's see what we have. And the first question was, is our modification impairing this SNH3? So here is Arabidopsis for comparison. We're looking at nuclei in leaves, and we have the wild type. The centromeres are occupied by SNH3. And here are two good haploid inducers, and we see the same, no difference. Well, in Camelina, situation is different. We see it in wild type, 20 chromosome, and then in the two haploid inducer, even in leaves, where we would expect to see this, there's very little SNH3 or none at all. In cell division, very little SNH3. So we certainly have impaired, and I'm not going to take you through this, but essentially shows that SNH3 is localized on centromeres very efficiently in a root division, and here you can hardly see it. So we have impaired SNH3 efficiently because we see very little of it. And then what are the consequences? Well, that's in progress. Let me take you to the next question, which is, are the centromeres of Hispida different? And also one expectation of the Malik-Hennikov hypothesis is that a repeat that binds SNH3 more efficiently has the potential of spreading through the genome rapidly and uniformly. Has the potential, I make the point. So what do we know about centromeres? Typically, centromeres have repeats, although that's not necessarily true for every centromere. But in most centromeres, you find these uniform repeats. And new repeats are thought to be born from the core of the centromere and spread and eventually get pushed to the side, so that repeats which are older tend to be more differentiated and flank the core centromere where SNH3 is bound and the kinetochore forms. Now this happens apparently centromere by centromeres, which means that they evolve independently of each other and form distinct centromeric haplotypes called synapse. And here's human genome. You don't see clearly what I'm pointing out, but there are centromeric haplotype. These were described by my colleague Chuck Langley and his lab at Davis. And that's what they form. It tells you that those repeats have been sitting on those centromeres for a long time. And we know that Arabidopsis does the same. For example, here's some data from Shamoni Maheshwari in my lab, where she actually identified a type of repeat that was SNH3 bound and was specific for one centromere only. There's two signals here. But more impressively, the recent studies on T2T sequencing have revealed very clearly that the centromere structures in different variety of Arabidopsis is different in the arrangement of the repeats, different in some of the sequences and some of the SNPs that are present. So old centromeres. And did I get blocked? No. So we wanted to identify these synapses, and one way to do it is to, in Camelina, is to sequence T2T. But we actually have another method that I'll describe here. You take a population that is genetically different, such as a backcross population here, and so you have centromeric regions that are either Columbia or Landsberg erecta here in this population. So if you have a map of those, you essentially have the solution to identify the synapse. You genotype these by common genotypic markers, and then you extract k-mers from k-mers are a little snippet of sequence, and you extract high copy number k-mers, which have to be some type of repeats. Then what you do, you associate them, and you generate a matrix, and if you have synapse, you form these distinct blocks that correspond to each of the 5 centromeres. That tells me that we have in Landsberg erecta, we have sets of repeats that are unique to chromosome 4, unique to chromosome 3, and so on and so forth. So this is a simple, rapid way of extracting centromeric repeats. And we can do it for Cole, of course, in the same cross. So now we did it for Camelina. The genome of Camelinas are quite distinct, the Hispida from the Neglecta. So you take the Hispida, you start with the neglecta repeats and ask the same question. Can I find the synapse? And the answer is yes, they come up readily. Here are the synapse on the neglecta genome. And I'm going to skip through some of the detail here, but if you do it for Hispida, you don't find any synapse. That is, each centromere of Hispida, shown here in green, when you use the Hispida k-mers, hardly shows any differentiation, which tells you that these repeats are much younger. So the situation here is, I can turn the, oops, it's not moving forward. Any computer savvy person here who knows how to, oh, here it goes. So so the conclusion is that that in neglecta, we have deep phylogenetic distances, so to say, and in Hispida, on the other hand, we don't, or at least we think that the evidence is consistent with there having been a recent expansion. So this is where we are. We are really interested in this recent expansion and the possibility that this will have been caused effectively by a higher efficiency of interaction with SNH3. So in summary, the translating this technology from Arabidopsis to other species is challenging. It can be done, and Camelina shows you that it's not simple. Clearly we can compromise SNH3 function, but the result that you get may vary a little bit. We're trying different ways, by the way, to get a perfect haploid inducer, so we're not yet throwing our hat or calling ourselves out of the fight. But we're happy that we found this surprising genomic bias because it allows us to test interesting hypotheses. I told you about the two explanations, and we think that the Hispida may have, in fact, stronger centromeres. So what about then moving this technology in other crops? So the problem, we think, is that this protein evolves fast, and so the interactions with other subunit can fall under slightly different rules in the affinity, in what happens. So you have to find a sweet spot. I have no doubt that there is a sweet spot in every crop. The challenge is to identify the sweet spot. And you know, if you were in the process of doing this, I would say steer away from the GFP tail swap, because GFP tail swap is very severe. It works well in Arabidopsis, but it's too severe for many other species. I would use predicted intermediate alleles and try to test a range of SNH3 alleles that you think may give you a phenotype. The heterozygous SNH3 is worth trying, and I would recommend trying to set up your CRISPR in such a way that you can mutagenize only one SNH3 and leave it heterozygote. So you would make a hybrid and then play on a polymorphism. We actually done that. It's possible. So you get a clean heterozygote. Then you complement it after that. And there are labs that have developed targeted proteolytic removal in the egg cell. The Huben lab has a paper in bioRxiv. And so that's an interesting technology. And you know, essentially we have to keep going. And I acknowledge all my collaborators and the funding. This is my cousin, Claudio, and he is actually in Venice, where he lives. This is not a gondolier. He has his own gondola-like boat, and he goes shopping with it or something like that. I want to leave you with that image, symbolic of the fact that we have to keep rowing and go toward the good things. Thank you.
Thank you, Dr. Comai.